National Science Week is an initiative of the Australian Government. It's aimed at increasing participation in science and inspiring a next generation of participants in science. A collaboration of the Manning River Turtle Group, the University of Canberra, Charles Sturt University and MidCoast Council applied for a Science Week grant and were successful in receiving that grant. The grant is for engaging with schools and environmental organisations to learn about the process of eDNA sampling, and then the organisations involved will go to a site, they will take their samples and we will analyse those samples and talk about the results.
There's a really great collaboration between all stakeholders in our area, so that's between council and LLS and national parks, and then community organisations as well, which are all working together to enhance our knowledge.
So eDNA stands for environmental DNA, and it involves us trying to detect the DNA that all organisms shed into the environment. So for some animals it might be scales, like fish will be constantly shedding scales into the environment. Birds are shedding feathers or skin cells, or mammals are going to be shedding fur into the environment. So we can go in and try to detect those bits of DNA by taking a water sample. Incredibly sensitive way of detecting species. It's also non-invasive, so it just involves taking a simple water sample and doesn't involve any interaction with the species. So really great for detecting endangered species, for instance, and a whole lot of rare and cryptic species that might be very difficult to detect using traditional methods. We're going to look at the little segments of DNA, which we call DNA barcodes, and that helps us to identify which species has left those bits of DNA behind. So that can give us an idea of the species that are present within a particular environment. And that might be anything from the fish that are fully aquatic, it might be semi-aquatic species like platypus, or it might even be terrestrial species like dogs or birds or bats. So for the Manning River turtle, we're developing a species-specific primer. So what that involves is looking at the DNA of the Manning River turtle and finding a location on that that's unique to the Manning River turtle. And we're going to go in and target that from the water samples that we take. So that will help us to really hone in on the locations of the Manning River turtle when we sample out this catchment.
Council benefits greatly from these kinds of projects, and eDNA being interesting and new brings in community members who want to get involved in science, and science is a really good way to link in with community.
We'll be able to compare different sites with the different animals that we find there. We'll be able to establish a baseline for future rounds of monitoring to see if there's an increase in diversity or a decline.
The process is actually surprisingly simple, as long as you're careful with not contaminating the filters, but it's really simple. So I'm really hoping that we'll get a lot of data out of it, and then it's going to be something that a lot of people might put their hands up to do.
It's like CSI for the environment, you know. To think that some animal, you know, maybe 100 metres or a kilometre away upstream can be doing its thing in the water, and down here we take a water sample and bring that back, send it off to their lab, and they can show us that it's there. It's fantastic.
So we're going to be looking around the Copeland area, and we're particularly interested in, hoping to detect some of our more threatened frog species, like the stuttering frog. I also know that Copeland's a bit of a hotspot for koalas too, so eDNA is pretty cool because you can even detect remnants of terrestrial species, so it'd be cool to see some of those species.
These types of programs, so the eDNA program, and getting people out into to actually be involved with that learning and teaching, and spreads the word so people get a better understanding, have ownership, and then you actually might protect and conserve.
We do a lot of camera trapping, but sometimes we don't always get the animals on those cameras. So through scat analysis and things like eDNA, that can just further support all our findings, all our data. We can now incorporate this into all our programs.
I really like eDNA because it's a good way to see what's in the water without harming the animals.
I'm hoping to find some platypus, Manning River turtle and the rakali, which is a water rat.
The eDNA process is just another tool in our data set and resources to enhance our growing knowledge base of such a special area.